Hello everyone and welcome to lecture 16. We're going to talk a little about electrolytes and acid-base regulation. Okay, so the electrolytes that we're going to cover in today's lecture, we're going to talk about sodium regulation, potassium, hydrogen ions, and bicarb. And then we'll talk about, of course, how the hydrogen ions and the bicarb, how they're regulated to help maintain our pH in our blood. Okay, so to start with sodium regulation, all right, so first off, we reabsorb, throughout the day we reabsorb most of our sodium. And it's the reabsorption process of sodium is the major energy requirement of the kidney. In other words, a lot of the transport mechanisms we use to transport sodium from inside the nephron lumen back into the blood requires a lot of ATP. And so that's really its major energy requirement. So if you recall from the first lecture, I talked about how we'll move sodium out of the lumen of the nephron and into the blood, and with it, we'll also take, let's say, it'll co-transport glucose or amino acids or phosphate or something like that. Or we could have, anti-transport where sodium is being brought in and we're secreting, hydrogen ions into the lumen. And so sodium is coupled to the movement of a lot of these different substrates. All right, so as I said here, glucose co-transports most sides like glucose, phosphate, and so on. All right, as well as the movement of sodium also influences the movement of water in many parts of the nephron, as we discussed in the previous lecture. Okay, so it can establish an osmotic gradient for water flow. And of course, secretion of proton ions, which we'll talk about in more detail today during this lecture. In terms of maintaining sodium balance, okay, just in general, if we keep in mind from last lecture, all right, if we move sodium ions, water tends to follow it, at least in certain parts of the nephron. And so ultimately, sodium gain, meaning reabsorption of sodium, leads to an increase in the extracellular fluid volume because water will typically follow that sodium. All right, and in fact, in areas of the nephron where it doesn't directly follow it as we reabsorb it, instead it's used to establish a very high concentration in the interstitium, like in the medulla, wherein we can actually reabsorb water somewhere else in the nephron. So again, we use it to help draw water into our bodies. And if we have any kind of sodium loss in the urine where it's being excreted, that's ultimately going to lead to a decrease in our volume. And so, regulating sodium levels also helps to regulate our water volume, or our ECF volume, excuse me. Okay, so let's take a look at how sodium is reabsorbed in different parts of the nephron. So in number one, we have sodium reabsorption in the proximal convoluted tubule. So that's just after it gets filtered in. All right, with the rest of the solutes, it's going to move out of the lumen. So over here in the drawing is the lumen. All right, over here on the opposite side is the blood. And of course, these would be the cells lining the nephron. And on what we call the apical surface is the surface of the cell that faces into the nephron lumen. And you'll see sodium can be reabsorbed, and when sodium is being moved into the cell, it's being co-transported with glucose, amino acids, and phosphates, as I've mentioned before. So it helps to move those inside the cell down its gradient. All right, and sodium can also move inside the cell and transport or secrete protons out into the lumen. All right, so we'll keep that in mind. Now, again, as sodium moves inside those proximal tubule cells, it will start to accumulate and we won't have as much driving force for sodium to move inside the cell. So to help counter that issue, we have on what we call the basolateral sides. So the basolateral sides of the cells are the ones that face the interstitium and the capillaries. And so what you see here is the sodium-potassium pump. So it moves the sodium out of the cell and back into the blood in exchange for potassium. And then we'll have some channels on the basolateral side that can help potassium also leak out so it doesn't accumulate too much either. And so in this regard, what we're doing is we're removing that sodium that's coming inside the cell. This creates or keeps the gradient so that sodium on the luminal side will continue to want to move into the cell. And as it's moving into the cell, it'll then get pumped out of the basolateral side and back into the blood. So this maintains that gradient, so we still have that driving force, which helps us to pull our solutes in. Number two, we also have the reabsorption in the loop of Henle. So just as a reminder, sodium is, it's about 67% reabsorbed in the proximal convoluted tubule. In the loop of Henle, All right, we're going to about 25%. So here, 25%. And again, in the ascending, the thick ascending loop of Henle, remember there's impermeable to water, but we do have on the apical side, we have this transporter here that's moving sodium, chloride, and potassium, ultimately to fill it, to be distributed into the interstitial space of the renal medulla, okay, to create a really large concentration gradient so that we can have a nice large gradient to reabsorb water from the collecting duct, for example. All right, again, you see these sodium potassium pumps on the basolateral side, again, to help maintain that gradient inside that cell. Over here, number 3, this is in the distal convoluted tubule. And one of the primary transporters there is a co-transporter with sodium and chloride. All right, sodium and chloride. Same thing, you'll see some channels on the basolateral side for chloride, so that can escape into the interstitium where the capillary is there, and we also have our pump there. So again, you're going to see these active pumps, and again, this is one of the major energy requirements, is just the ATP requirement to keep these pumps working, to maintain those gradients, so that they can constantly be pulling sodium in, sodium chloride, and some of these other ions as well that I've mentioned. Now in the distal convoluted tubule, you're looking at maybe another 5%. All right, the only part I haven't shown you right here is the collecting duct, only because the collecting duct is going to have specific cell types that we're going to have more control over in terms of hormonal control. Up until this point, the proximal convoluted tubule, the loop of Henle, and at least the early part of the distal convoluted tubule are fairly fixed in terms of how they operate. We can ultimately affect them with hormones by increasing or decreasing their amount of reabsorption, but they're all essentially reabsorbed here. Once we get to the collecting duct, we get a little bit more regulatory controls. We'll talk about that. And so the final 3% of reabsorption can take place in the collecting duct, depending on how these cells are responding to hormonal influences. Okay, so let's talk about, at least broadly, how some of the factors affect sodium excretion. All right. Again, we're going to maintain sodium reabsorption in, say, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule at those percentages, 67%, 25%, 5%, and so on. Where it becomes a little bit iffy in terms of our reabsorption becomes the late distal convoluted tubule and the collecting duct, because that's where we have more variability, and we can influence those cells to, increase or decrease their ability to reabsorb sodium. And so that's really where most of the effectiveness comes in, if we're going to excrete sodium or not. But there's other factors that play a role in determining essentially how much can get excreted in terms of total amount. Okay, in terms of percentages, we're still going to reabsorb the same percentages. So for example, GFR, right, if it increases, we're going to increase the filtered load of sodium that gets into the nephron over time. And along with that, we'll still reabsorb the same fraction of sodium out of that, all right, all along the nephron. And that's because the reabsorption rate will match the GFR. Okay, so we'll still reabsorb that. Now, when we get to the late parts of the nephron, if there's still a large sodium load and high volumes, we can still alter the amount we reabsorb, depending on the hormones that are activated. Now, and speaking of the hormones, we have aldosterone. So aldosterone works in that late distal convoluted tubule and the collecting duct. Now that's, again, if you remember, this is from the last lecture. This is primarily the area of the nephron that's under the most influence when it comes to some of these hormones, okay, and how we regulate volume, for instance, in the last lecture. Here it's how we regulate sodium reabsorption, all right? Because otherwise, up until that point, it's almost an automated type procedure. And again, these hormones that can alter some of that reabsorption, of course, all right. But the most of it's going to happen here at the distal area. So aldosterone, and we'll talk more in detail about that, which will cause, in brief, just sodium reabsorption. Atrial natriuretic peptide, or ANP. ANP has effects that are very broad across the whole nephron, as well as the vasculature, that the peritubular capillaries, like the vasa recta, for example. It can also change its properties. And its main goal here is to actually reduce sodium reabsorption. If I reduce sodium reabsorption, that's going to cause an increase in the excretion of sodium, and water will follow that, and I can reduce the volume. Okay. Sympathetic nervous system. The sympathetic nervous system, broadly speaking, it'll increase sodium reabsorption. Okay. So it can actually directly influence proximal convoluted tubule cells to increase sodium reabsorption. And we have another example like insulin. So insulin has also been shown to increase sodium reabsorption as well. So many times we'll have this sort of concoction of hormones that can increase our reabsorption, and if there are low levels of these hormones, then we will end up excreting more of them. Obviously, with the obvious exception being A and P. Now, of those hormones, aldosterone is one of the main ones that we're going to be talking quite a bit about. So this again acts on the late distal tubule and the collecting duct. All right, the two types of cells that of interest in that area are the principal cells. All right, so the principal cells and the alpha intercalated cells. There are different subtypes, alpha, beta, and so on. Mostly we're talking just about the alpha and beta intercalated cells. So in terms of aldosterone's actions, these two cells are found only in that late distal convoluted tubule and in the collecting duct. And so you can see over here in the drawing, all right, here's the aldosterone coming from the blood. And again, remember the aldosterone is released in response to angiotensin II. It's released from the adrenal gland. And aldosterone, all right, what it's going to do is it's going to act on the basolateral side. It's going to act by enhancing the sodium-potassium pump. It's going to increase the number of channels on the apical side for sodium and increase the number of potassium channels on the apical side. Remember, the apical side is the side that faces the lumen. Ultimately, what's happening is this. When aldosterone increases the number of sodium channels on the surface, that's going to allow for more sodium to go from the lumen inside the cell. So it's going to increase the diffusion there. It's also going to allow potassium, which is in higher concentrations inside the cell, it's going to allow more of that to diffuse out by inserting more of those apical channels. And by increasing the activity of the sodium-potassium pump down here, that's going to help maintain that gradient so that we can really pull that sodium out of the lumen. At the same time, we'll also lose more of the potassium. And then, of course, there'll be some leak channels here on the basolateral side, but the apical side is where the main effect comes with aldosterone. So, in summary, aldosterone causes an increase in reabsorption of sodium and secretion of potassium. And by reabsorbing the sodium, water is going to follow with that. So we got sodium and water reabsorption, we secrete potassium, and this is all occurring in the principal cells based on the mechanisms I just talked about. It also has an effect on something called the alpha intercalated cells. So when its levels rise, not only does it influence the principal cells, it'll act on the alpha intercalated cells and stimulate those cells to secrete protons or to secrete that hydrogen ion. And this is a proton ATPase. So it increases the activity of this ATPase, so it starts to secrete more of these hydrogen ions into the lumen. The net effect is at high levels of aldosterone, you'll reabsorb sodium and water and increase extracellular fluid volume. You'll also be losing potassium because you're secreting that into the lumen, all right, and which could ultimately be excreted. So it's going to secrete it and excrete that potassium. So we're going to have a drop in the potassium levels, and we're also going to be secreting protons, and these protons will also get excreted. So in effect, you can increase your sodium, decrease your potassium, and you could also increase your pH by getting rid of some of those protons. Okay, so in this cartoon, there's a couple things I just want to point out. I'm not going to cover everything that I've drawn here, just a few features that I just mentioned. So first off, just to orient yourselves, down the center here is the lumen. So that's the lumen of the nephron. There's cells on either side. So you can see I've written one out as principal cells. The other one's alpha intercalated, and I also put in a beta intercalated cell there. On the outside of the nephron, you can see in red on either side, those are the peritubular capillaries. Okay, so that's just to help us orient. Now, in terms of aldosterone's actions, remember this is in the late distal convoluted tubule and collecting duct. And it's going to act on principal cells to increase the number of potassium channels on the apical surface. All right, as well as, which I forgot to draw in here, but I'll draw it now, as well as increasing the number of sodium channels on the apical surface and the activity of the sodium-potassium pump on the basolateral side. So it creates that basolateral sodium-potassium pump, will create the large gradient necessary to drive sodium into the cell and potassium out of the cell and into the lumen. And then over here, the alpha intercalated is the one I mentioned before. It's got multiple functions which we'll review in a later time in this lecture, but for now you'll see that on the apical side of the alpha intercalated cell is an ATPase that secretes protons, and its function is greatly enhanced by aldosterone, so it'll secrete more protons. The net effect in terms of what happens to the body in terms of its electrolytes, because of the level of aldosterone, if it goes up, is you're going to increase your ECF volume, because as it reabsorbs that sodium, water is going to follow it. The potassium levels are going to drop because potassium is being secreted at the same time, and your proton levels are going down, which means your pH is going up, so the person can become alkalotic. So you can imagine under normal circumstances they wouldn't become alkalotic, for example, but if say it was elevated in a condition like hyperaldosteronism, then you could see sort of the extremes of that, which would be, hypokalemia, increased volume, and alkalosis, for example. All right. So in terms of mechanisms for this reabsorption, all right, in general, all right, too much sodium reabsorption, all right, it's going to result in an increase in our ECF volume, because again, it's an osmotic agent. When it's reabsorbed, the water follows it typically, right? So that can cause volume overload if there's too much of that reabsorption. So if we do have volume overload, all right, and not necessarily because we've reabsorbed too much sodium, maybe we've ingested too much volume and so on. What is the renal's response to an increase in ECF volume? Well, it's going to increase the arterial blood pressure, because some of that fluid, as we've learned from the first couple of lectures, distributes to the vasculature, all right, as well as the interstitial space and maybe the cells, depending on its tonicity. Now, of some of that volume, that's going to increase the hydrostatic pressure, which is going to be an increase in our blood pressure. What that's going to do is stretch the afferent arteriole. All right, the afferent arteriole is that beginning portion that goes into right before the start of the glomerular capillary. And so here we have these specialized juxtaglomerular cells, also known as granular cells. And so it stretches these afferent arterioles because we have a surge in the volume. And that stretch is going to cause a reduce in renin release. So if we have a large volume, we stretch those cells, they reduce their renin release, which means we drop our angiotensin II levels, which subsequently is going to result in a drop in our aldosterone levels as well. So the net effect then is, by dropping those hormones, we don't vasoconstrict as much, and we won't reabsorb as much sodium and water with it. So we can help reduce some of that volume. So this will reduce the reabsorption, for example, from the principal cells, right, located in that distal nephron. Now, if we have an increased GFR, all right, because let's say we have a larger volume, it's increased our GFR. So what ends up happening is we have an increase in our sodium chloride that reaches that macula densa that I talked about in the first lecture. The macula densa cells, all right, they're going to signal the juxtaglomerular or granular cells, and it's going to decrease their release of renin. So there's two mechanisms. We can stretch the afferent arteriole. That's going to reduce renin release. We also have an increase in our GFR due to our rise in pressures, and that's going to, the macula densa then is going to signal those granular cells, and they're going to reduce aldosterone in response to that signal, and so same net effect. Okay. Finally, if you have a large volume, you can stretch the atria of the heart, and they'll release that AMP, so atrial natriuretic peptide, which I mentioned a few slides ago. And AMP's actions on the nephron is to reduce sodium reabsorption. So we'll excrete more sodium, and volume will follow with that. And stretched baroreceptors, so an increase in pressure, as you guys know, signals the baroreceptor, or changes the rate of firing of the baroreceptors, excuse me. And so that's going to reduce sympathetic activation, right? Increases the signal, sympathetic activation goes down, and the sympathetic nervous system can directly affect sodium reabsorption in the nephron, so it reduces its ability to reabsorb some of that sodium. Now, too little sodium reabsorption is going to result in a drop in our ECF volume, okay? And so it's going to be the opposite of all the things I just talked about. So, for example, you're not going to stretch the afferent arteriole. Renin is going to be released in higher concentrations. You're going to have a decrease in the sodium chloride that reaches the macula densa, and that's ultimately going to influence an increase in renin release. And you're not stretching the atria, so AMP levels are going to drop, and the baroreceptors, okay, they're going to decrease their firing rate as the pressures drop, and the sympathetic activity is going to increase. And so these are all the opposite effects that will help to reabsorb more sodium in a state where there's low volume. Okay, so now I want to talk about regulating the potassium levels. All right, now when we talked about sodium, sodium was primarily just reabsorbed. I mean, we don't secrete sodium. We just regulate the amount that we actually reabsorb, and then we leave the rest in the lumen, which can have effects on things like volume that we excrete ultimately as well. But potassium, on the other hand, potassium can actually be secreted and reabsorbed. So it's one of those few electrolytes that can do that. And so what we're going to have is a balance between secretion and reabsorption. And so this can be influenced by several factors, which we're going to talk about in the following slides, things like diet and pH, and so on. So primarily, just like we saw with sodium, potassium gets primarily reabsorbed in the first half of the nephron. Okay, so what we're going to see is just reabsorption in the proximal convoluted tubule, just reabsorption in the loop of Henle, and the percentages are effectively the same, like 67% in the proximal convoluted tubule, and another 25% in the loop of Henle. And that's only just reabsorption in the first half there. But once we get to the distal convoluted tubule and collecting duct, then we can toggle between secretion and reabsorption. All right. And net secretion can occur when the dietary intake of potassium is high. So if we exceed our potassium levels that we need, we'll just excrete that. So the potassium levels will become high intracellularly, okay, and we'll secrete that and ultimately excrete that into the urine. Otherwise, if the potassium, let's say dietary potassium levels are low, we'll typically reabsorb the potassium to maintain our concentrations. So just to kind of emphasize the importance of potassium regulation, you guys remember from early on, you know that excessive potassium levels can lead to things like dysrhythmias or arrhythmias, and we talked about that when we talked about how it can cause changes in the driving force of potassium, for example, which can change the resting membrane potential, right? So these can actually be quite deadly if they're dysregulated. So what ends up happening is we have several mechanisms in play so that when we ingest large loads of potassium, which our typical diets far exceed what we need, we need to make sure that first off, we put them inside the cells and they don't stay in the extracellular space for too long because we don't want to have a large imbalance. And then once we have that large accumulation inside the cells, like in the collecting duct cells, for example, we want to make sure that we offload a lot of that high potassium level into the lumen and excrete it. Okay, so we can readjust or keep our potassium levels within a healthy range. Okay, so most times with standard diets where we spend more time secreting than reabsorbing. But certain diets, potassium levels might be low, and so therefore we will reabsorb them. Okay, so in terms of the factors that are going to influence potassium reabsorption or secretion in that late nephron, one of the ones I mentioned already was dietary intake. So again, just broadly and logically speaking, if we have an increase in our dietary intake of potassium and we exceed the amount necessary, all right, then what's going to happen is that it's going to be increase the amount of potassium that gets taken up into our cells, so that concentration increases. And when that happens in the cells lining the nephron, that's going to result in an increase in the secretion. All right, and so here I'll draw that out for you. So we're looking at a high potassium diet secreting through principal cells. So if I draw the principal cell right here, we'll say this is the lumen. So we know that this is the apical side. And this is our principal cell. Here's the capillary, and I'm going to draw, this is my sodium potassium pump on the basolateral membrane. That's the sodium moving out, the potassium moving in. And on the apical side, I'm going to draw some potassium channels. Okay, so we saw this was the cell that aldosterone acts on, okay? But even without aldosterone, this will work. So what happens is this. Let me draw here. So what you can see is this. If we have an increase in our potassium concentration, all right, so we have an increase in potassium concentration. As the blood flow goes through these capillaries, okay, one, you're going to get a net, remembering that in the beginning parts of the nephron, we're just reabsorbing potassium, so we're going to have an increase in our filtered load at a higher potassium concentration. And the same percentage is going to get reabsorbed regardless of that in the proximal convoluted tubule and the loop of Henle. But here in the late areas of the nephron, this is where these cells can have more of an impact in terms of making sure we can offload more of that potassium if needed. So if there's a high concentration here in these capillaries, that's going to increase the activity of our sodium-potassium pumps, and we're going to get an increase in the intracellular amounts of potassium. That large increase in potassium actually increases the driving force for potassium to leave the cell. So here we're going to have channels on the apical surface. So if I have a larger concentration, I'm creating a larger gradient, and I have channels on the apical surface. This is going to allow them to really flow down that concentration gradient. So it's increasing what we call that driving force, if you remember. So with high diets, we end up with high intracellular potassium concentrations, which means we can actually secrete more potassium. Now, the beauty of the design is that, if the potassium concentration is low, all right, so I'll get rid of this. If the potassium concentration is decreased, on the other hand, I don't have as much potassium, let's say, inside the cell, and my driving force becomes less. So I'm not going to secrete as much potassium. So it's proportional, all right? The concentration of potassium goes up, I'm going to secrete more through these principal cells. If it goes down, I'm going to secrete less. So it's a nice sort of direct relationship that they have. Now, surrounding these principal cells, I also have those alpha intercalated cells, for example. Let's put the capillaries over here. All right. Now, on the apical side here, I'm going to draw this exchanger. I also have this mechanism. Now, this mechanism is going to be useful in states of low potassium diet. So in low, we already know I'm not going to be secreting as much simply because I've reduced the driving force out of those principal cells. But at the same token, all right, I also have these intercalated cells that can also do an additional job. And so what happens there is, on the apical side of the alpha intercalated cells, they have a proton potassium exchanger. So what this does is this brings potassium in exchange for a proton into the lumen. And this is an ATPase. And so states of low potassium concentrations, all right, in the lumen, so low potassium concentrations in the lumen of the nephron here, this actually stimulates. So low potassium in the lumen, which is going to come into contact with this apical exchanger, that low concentration actually enhances the activity of the ATPase. So what ends up happening is you actually start to reabsorb more potassium here. So in this fact, you reabsorb more potassium, but you're also reabsorbing it with exchange for a proton. So this is where you're going to see this link between potassium and hydrogen ions. We saw it briefly actually when we talked about aldosterone, because aldosterone's effect was to reabsorb sodium in water, secrete potassium, as well as secrete a proton. Here you're seeing potassium when it's being reabsorbed, okay, because let's say we had a low diet, so it's a low filtered load, that stimulates these apical exchangers, and it's going to secrete protons. So this is how hypokalemia, or a state of low potassium, can cause alkalosis. Because in a state of hypokalemia, depending on how low it is, we're going to reabsorb more potassium, and we're going to excrete protons in exchange for that, and so that can make the person alkalotic. Okay, so there's other factors that influence potassium secretion and absorption in the distal portions of the nephron. So we talked about dietary being high dietary intake or low dietary intake. And when we talk about diet, we said that aldosterone, in cases of high dietary potassium intake, that can stimulate aldosterone. And in fact, plasma potassium concentrations, if they're increased, can actually stimulate the release of aldosterone, because aldosterone is one of our main regulators of potassium secretion. So when aldosterone levels go high, potassium secretion increases. And it does so by influencing those principal cells, activating those sodium-potassium pumps. So we get a high potassium concentration inside the cell. That leads to a large driving force of potassium out of the cell through the apical side, through those open channels, those potassium channels that face the lumen. So this way we can secrete more. Okay. Now, aldosterone has a very large influence on our regulation of potassium secretion, and in patients who have low aldosterone levels, their potassium levels could be somewhat dysregulated and actually be elevated. Rate of flow can also influence potassium secretion. And so if I have, let's say, my principal cell here, so we'll call this the P cell, principal cell. And let's say this is the lumen over here. And I'm going to put on the apical surface some potassium channels, and this is, we have a high amount of potassium inside these principal cells thanks to the sodium potassium pump. And let's say in the lumen I have an increased flow rate. Now, an increased flow rate, this is the volume that's moving past these principal cells over time. Now, if I have, an increased flow rate, usually that means that I have an increased volume, probably due to, not reabsorbing sodium, so I have more water there, and so I increase my flow that way. Or somebody is using a diuretic, like a loop diuretic, or thiazide diuretic, and that's going to increase the volume that's inside the distal nephron there, and it also increases the flow rate past these cells. And so the effect is that if I have an increase in flow rate, I'm also going to get an increase in my potassium secretion. And the reason is this. As I have a high concentration of potassium in those cells, they naturally want to go, their driving force is to be out of the cell. And if the flow rate is very slow, right, there can be some potassium accumulation just outside the cell in the lumen there, and that can limit the driving force, at least until the solution moves by. The faster that solution moves by, the more it essentially washes out what has accumulated there, okay? So as potassium moves down its gradient and into the lumen, that's getting washed out by this continually fast-moving flow. And so what ends up happening is it creates and kind of maintains a large gradient there. So potassium is constantly going to be moving down and into that lumen with a larger driving force. So in summary, though, an increase in flow in the lumen there causes a larger secretion of potassium, a decrease in flow, a decrease in the secretion. And the benefit of having this particular setup is that in some cases, remember, aldosterone is one of our main regulators for potassium secretion. But if we have a lot of sodium and we want to get rid of that sodium, okay, with high sodium and high volume, that's going to inhibit the amount of aldosterone that we actually are producing. So aldosterone levels would be low. They're not going to be reabsorbing that sodium and water, and that's going to increase flow. The problem with it is if aldosterone levels aren't high and I'm not reabsorbing the sodium because I'm trying to excrete it, that also has an effect on my ability to secrete my potassium because they're linked, right? Because remember, aldosterone reabsorbs sodium and secretes potassium simultaneously. So in this case, by not reabsorbing the sodium and aldosterone levels are low, it's going to increase my flow. So in this way, I can still secrete potassium, all right, even when aldosterone levels are low, by having an increase in my flow. All right, due to, again, the high volume that'll eventually be excreted. In addition, because potassium is positively charged, it'll more likely move into the lumen and down its concentration gradient if there's a big charge difference. So for example, if I have something like a lot of bicarb that happens to make it into my distal portion of my nephron there, bicarb is negatively charged, potassium would be attracted to that, that'll increase the driving force. So if I have some sort of organic acids or what have you that have made it that far into the nephron, that will attract and pull out some of the potassium as well. Last note, just a clinical one. When it comes to the rate of flow, when we talk about use of diuretics, one of the side effects of diuretics is a loss of potassium. So they can become hypokalemic. So we have to be careful with things like loop diuretics and thiazide diuretics because it does increase flow. The increased flow causes an increased secretion of potassium. So we have to monitor potassium levels. And in fact, they've got diuretics, for example, that are called potassium-sparing diuretics. And we'll get more into that later on, but the whole idea with that is that simply we're increasing flow, which increases that secretion. Okay, so the last regulator of our potassium secretion has to do with our acid-base balance. And this is related to these proton-potassium exchangers, one of which I already mentioned, which was on the apical side of our alpha intercalated cells. In a state of hypokalemia, for example, we reabsorb the potassium and we secrete the proton. By secreting the proton, we can actually, elevate our pH and become alkalotic. So down here, clinically speaking, yeah, hypokalemia can cause an alkalosis type state. So we mentioned that. Now here, whether our blood is acidotic or alkalotic can change the level to which we regulate secretion of our potassium. So let's take an example. On the basolateral surface, we can also have these exchangers. So I'm going to say the lumen is over here. Here's the lumen. We'll just draw the capillary on this side so we can orient ourselves. We have our potassium, apical potassium channels. I'm talking about the principal cell here. This is the principal cell. And then down here, I'm going to draw our exchanger, our proton potassium exchanger. That's what happens in this case. Let's take acidosis, for example. So we'll take this first case here where acidosis can cause hyperkalemia. In a state of acidosis, we have an increase in our proton concentration. What this means, all right, is that these exchangers, what they'll do is they'll move the proton into the cell in exchange for the potassium to help us regulate our pH there. So that's going to move the potassium out. So because of these exchanges, and these exchanges are located on these principal cells as well as on the basolateral side of many of the different cells, including things like the alpha intercalated cells. But what happens is the proton then moves, the high proton concentration moves inside the cell, and it moves the potassium out of the cell and back into the bloodstream. So what ends up happening is the potassium concentration goes up out here. That potassium concentration starts to increase. So that's effectively causing our hyperkalemia, and the protons are moving in. Now, how that affects potassium secretion is for this reason, is we're moving potassium out of the inside of a cell here in exchange for the proton, so the potassium concentration is going to go down. If the potassium concentration goes down, that's going to reduce, the driving force for potassium out of the cell and into the lumen there. So this is how acidosis can cause a hyperkalemia. We're not secreting as much, and we're replacing those protons with potassium in the blood. The reverse is true for alkalosis. So for alkalosis, I'm going to get rid of this here. So for alkalosis, we have a drop in our proton concentration, which means our pH is going up. The exchanger will not be moving protons in any longer. Instead, to counter that, it'll reverse. So it's a reversible exchanger. It'll bring potassium in exchange for the protons. So we're trying to bring the protons out of the cell in order to balance off that high pH and bring the pH back down. And it's going to load up the cell with potassium, so the potassium's concentration is going to go up, and it's going to decrease in the blood because we're removing potassium from the blood, and we're increasing it inside the cell there. And the effect, again, is the opposite of what we saw before. In a state of alkalosis, all right, as we try to pump those protons out and move potassium in, we increase the gradient, and we greatly increase the driving force to secrete potassium. So that can lead to hypokalemia. So in this case, alkalosis can cause hypokalemia. And so this is something you should look for. Anytime somebody is in an acidotic or an alkalotic state, their potassium levels can change because of these exchangers on the basolateral surface. And I already mentioned hypokalemia can cause alkalosis, and that was due to the apical exchanger. Hyperkalemia can also cause acidosis. So sort of the reverse of the acidosis causing hyperkalemia. And the mechanism of that, we're not going to get into in terms of its pathophysiology, but this is renal tubular acidosis can cause this. It has an effect on production of buffers. So you guys will, this beyond the scope of this lecture here, but understand that hyperkalemia can cause acidosis as well. So again, these exchangers are important, and they are affected by that, the pH of the blood. Okay, now bicarb and hydrogen ion are the last of the electrolytes that I want to discuss. And the renal regulation of this is obviously going to have a direct influence on acid-base. So after this, we'll go into some acid-base disorders and discuss those. So first off, when we metabolize, all right, our daily routine of just, living and eating and breathing, metabolic byproducts are acidotic or acids in nature, right? So like lactic acid, sulfuric acid, and phosphoric acid, and these are all products of catabolism. Breaking down phospholipids and breaking down proteins, we produce acids. And we actually produce an abundance of these acids. And so this would, just for normal metabolism's sake, would actually, plummet our pH if it wasn't for buffers. So we have a whole lot of buffers, and these buffers minimize the amount of those free protons because it's the free protons that dictate the pH. All right. So pH going up or down is based on the concentration of free protons. Buffers limit the amount of free protons available when they're produced, therefore preventing large shifts or changes in our pH. Okay. Now, what happens is when we regularly metabolize and produce those acids, we use up buffers. They all bind to those protons. All right. And so what happens is the unbound proportion of buffers decreases because they're binding some of these acids. So we lose a portion of those buffers. Okay. And so, the job of the kidney at that point is to eliminate the buffers that are bound to those protons and replace, replace some of the buffer that has been lost. Now, the kidneys can eliminate the excess proton, all right? It can get some rid of both free and ones that are bound to buffers, and it has to replace those used up buffers, all right, while reabsorbing the unused ones. So in other words, when we're metabolizing and our buffer levels are going down because they're binding to those protons, those will get excreted. But the buffers that aren't bound, because we metabolize so much and produce acids on a regular basis, we have to make sure that we reabsorb those buffers. All right, ones that are not used, we have to reabsorb those, and we also have to produce new buffers and get rid of any excess protons. So this is the job of the kidney now, is to make sure it can go through and do that, excrete the excess protons, reabsorb the unused buffer, okay, and maybe create some new buffer. Okay, so bicarb reabsorption is actually one of the major functions in our kidney to help us regulate our pH. And again, since we trend towards acidosis because we metabolize and produce acids on a regular basis, we have to make sure that any of the unused bicarb gets recycled. We have to reabsorb it. Okay, and we'll 100% reabsorb it in the nephrons under standard conditions. And 90% of that's going to occur in the proximal convoluted tubule, and the next 10% is going to happen by the time we reach the distal convoluted tubule. It'll all be reabsorbed. And this is all unused bicarb. Now, the method with which we can actually reabsorb this bicarb, all right, relies on the secretion of proton. So if this is the proximal convoluted tubule over here, all right, and we're filtering bicarb, so bicarb is in the lumen, I want to make sure I take that out of there and reabsorb that bicarb. So you can see some of that bicarb here. So if you recall, I secrete protons in the proximal convoluted tubule. I do that with the sodium proton exchanger. Remember, sodium goes inside the cell, protons get pumped out, and what happens is I actually can then combine the proton and the bicarb, becomes carbonic acid, and converts into CO2 and water. All right, right there in the lumen. And I can reabsorb that water, for example, and some of that CO2 can also go across those cells. And so once that enters inside the cell there, the CO2 and the water can recombine. Now the reason this happens, okay, in the lumen as well as inside these cells is because they have carbonic anhydrase. So just so we have a visual reference, CO2 plus H2O, all right, H2CO3, comes bicarb, HCO3 plus the proton, and here's our carbonic anhydrase. There's we'll call a brush border on top of these cells inside the lumen there, and that has carbonic anhydrase. Carbonic anhydrase is also located inside the cell. So it can make these conversions happen very rapidly. So again, as that proton gets secreted, it combines with the bicarb that got filtered. That converts into water and CO2, which can be taken up by the cells. That water and CO2 can then recombine inside the cell because we have carbonic anhydrase. And remember, it's a reversible reaction. And that can drive the formula in the reverse direction, where we're producing a proton and a bicarb inside the cell. The proton will get used to be resecreted, for example, back into the lumen to collect up more bicarb. The bicarb, on the other hand, that gets produced inside the cell will exit through a basolateral bicarb chloride exchanger. All right, so this bicarb will then get moved into the bloodstream. So it can't leave through the apical side. So what happened was this: I basically took bicarb that got filtered in, I converted it into CO2 and water, moved across the cell, I reconverted it back into bicarb and a proton. That proton got moved into the lumen, and the bicarb got moved into the blood. So in effect, this bicarb over here, I had to basically transform it into CO2 and water, transport it, and then convert it back. And so this is how we reabsorb this bicarb. This is not new bicarb in the sense I'm not producing new bicarb. I'm simply essentially reabsorbing the bicarb. Okay, and it relies on this protein secretion, which also relies on that sodium concentration to help pump the proton out. So They have the enzyme both in the lumen and in the cell to be able to carry this out. We can also get some of the CO2 from the blood there, which can help combine with the water inside the cell there and help us to make more proton if needed and to reabsorb more bicarb. So what I'll come back to, and this is just sort of alluding to this, is over here, the CO2 concentration in our blood, that partial pressure of CO2, can influence the rate and the amount with which we can reabsorb bicarb. Then I'll come back to that. So reabsorbed bicarb does not replace any of the bound bicarb that was used to buffer those free protons. We're simply just reabsorbing what was unbound. Okay, so things that can influence bicarb reabsorption. One is called the filtered load, which means the amount of bicarb that we're actually filtering into the nephron. The larger quantity of bicarb that gets filtered in makes it more difficult for us to reabsorb. So even though we reabsorb typically 90% to 100%, even within the proximal convoluted tubule, if the bicarb level is quite high, it exceeds our ability to reabsorb it adequately or at that same percentage. And so what ends up happening, some of that ends up getting past the proximal convoluted tubule, and most of that ends up not getting reabsorbed and gets excreted. And this is useful in cases where we have metabolic alkalosis, where the bicarb levels can be very, very high. We can excrete that simply because the filtered load is so great we can't really reabsorb as much of it. In terms of the PCO2 can influence the rate with which bicarb is reabsorbed. So this is my proximal convoluted tubule cell, and then here is my capillary. All right, remembering that we have carbonic anhydrase in these cells, and that can convert water, CO2, into carbonic acid, and then it produces a proton, right? So the PCO2 levels here in the blood can diffuse into the cell, combine with water, and ultimately to produce these hydrogen ions, and it can produce a high concentration of protons here, which can then go through the exchanger, right, and into the lumen there to combine with the carbonic acid, or excuse me, the bicarb, to be reabsorbed. If the PCO2 levels go up, that increases the concentration of protons that can then be secreted into the lumen, and I can reabsorb more bicarb. If the PCO2 levels go down, that's going to reduce the amount of proton I have there and reduce the amount that can get secreted, or the rate with which it can get secreted, and then my bicarb reabsorption will decrease. So again, an increase in PCO2 increases bicarb reabsorption. A decrease in PCO2 decreases bicarb reabsorption. This is useful in cases of respiratory disorders where if my PCO2 is high, right, so I'm not ventilating a lot, so my PCO2 is elevated, that can cause acidosis. So this helps me to increase my bicarb reabsorption in those states. And vice versa, if my PCO2 is decreased, I decrease the amount of bicarb that I can reabsorb, and that's useful in a state of alkalosis when I might want to actually be getting rid of some of that bicarb. And then finally, ECF volume. An increase in ECF volume decreases bicarb reabsorption. And then, an increase in ECF volume, so an increase in ECF, all right, decreases bicarb reabsorption. And a decrease in the ECF volume, something we also call volume contraction or dehydration, will increase bicarb reabsorption. So you can get something we call contraction alkalosis. So if you're dehydrated due to that low ECF volume, you can actually increase your reabsorption of. The mechanism for that, to explain that contraction alkalosis, which is something you guys can see clinically, is that, remember, when in a state of low ECF, you have an increase in your angiotensin 2 levels. That's supposed to be A2. So your angiotensin 2 levels. Angiotensin 2 can stimulate this exchanger, the sodium exchanger. Remember, sodium is moving in. It can actually increase the rate of sodium reabsorption here and proton secretion, which of course will increase the rate with which bicarb is reabsorbed in those states. Now we want to also excrete. So not only do we want to reabsorb the bicarb that was unused, we want to make sure that we excrete any excess hydrogen ions. And in doing so, we're actually going to generate new bicarb. So this isn't reabsorbed, it's new bicarb. And this happens in the distal nephron. All right, so we're talking about, again, the collecting ducts and late distal convoluted tubule. So bicarb has already been completely reabsorbed at this point. and our alpha intercalated cells. All right, so this is where they come into play. So over here we're looking at the alpha intercalated cells. So their job, all right, is to secrete a proton, and in the process generate a new bicarb. So the method with which it does it is that the alpha intercalated cells have, again, carbonic anhydrase. So we get CO2, right? CO2 combines with the water and produces a proton and bicarb. The bicarb has a channel, a chloride bicarb exchanger on the basolateral surface. So bicarb gets moved into the blood, and the proton gets secreted into the lumen. Now, this is exactly what we saw in the proximal convoluted tubule, with one big exception. The big exception is that in the distal tubules here, there is no bicarb in the lumen. There's no carbonic anhydrase in the brush border there. So when the proton gets excreted into the lumen there, it's going to bind to any buffers that are there that happen to be unbound. All right, so it's going to bind to buffers, and when the TA stands for a titratable acid, so we have buffers that we call titratable. One of the main ones is phosphate, so it can bind to phosphate, and another one is called ammonia. So we have the NH4, so this is produced from glutamine. So glutamine, the production from glutamine, excuse me, can produce NH3. NH3 in the lumen can combine with the proton and produce NH4+. NH4+ essentially traps that into the lumen. So basically this secreted proton is going to be excreted at this point. In the proximal convoluted tubule, the secreted proton does not get excreted because it's being used to recycle bicarb. The secreted proton here in the in the distal convoluted tubule and the collecting duct gets excreted because it's going to bind to these buffers, the titratable buffers like phosphates, and it's also going to bind to NH3. NH3 is probably the most important because the phosphates are going to be finite in amount, but our collecting duct here can produce NH3 as needed. And so the NH3 level, so for example, if we're more acidic, it'll produce more NH3 to help buffer the acids. So that'll produce NH4, and so the protons get buffered. There's very little free proton that ends up in the urine. It gets buffered, and primarily by these acids right here, by these buffers right here. And so these will get excreted. So these levels of titratable acid and ammonia, for example, those levels in your urine correlate with the amount of protons that you're excreting. Okay, so they get secreted, they bind to buffers, and they'll get excreted. Now the bicarb gets absorbed here into the blood. This is effectively new bicarb. It's new bicarb because there's no bicarb in the lumen. We're not going through that loop that we talked about in the proximal convoluted tubule. So for every proton we're excreting, we're replacing it with bicarb. So this is a major mechanism that the alpha intercalated cells use to create what we call new bicarb. Okay, and so in terms of those buffers, again, remember the binds to the buffers, the NH3 becomes NH4. We call this diffusion trapping. Once it becomes positively charged there, it'll stay in the lumen. And the production increases with a decrease in pH. So we'll produce more NH3 to buffer those protons. And in fact, the lowest our pH can go in our urine is about 4.5 or 4.4 because it gets buffered. There's very little free proton there. All right. Now the titratable acids, phosphates are probably the main one there. Now that'll make up about a third of the buffering, whereas the NH3 will make up about 2/3, half to 2/3. And depending on the state of acidosis, if we're more acidotic, more of it will be made. And so it'll make up a larger percentage of what's being buffered. And so that's an important feature that these cells have, is to be able to produce that buffer as needed depending on the acid state. So, in summary, in the proximal convoluted tubule, we reabsorb the bicarb via ultimately secreting protons to bring that to combine with the free bicarb, and then reconvert that in the cells and redistribute it. But in the late nephron, we're going to secrete proton, and there's no more bicarb in the lumen, so therefore it's going to bind to the buffers and be excreted. And then the bicarb that was produced gets moved from the basolateral side into the blood, so that's effectively new bicarb. Okay. So now I want to get more into the disorders, but I want to talk about this acid-base balance and talk a little bit about the chemistry behind it. Okay. So for example, the proton concentration, all right, that equals 0.00004 milliequivalents per liter. This is a very, very small concentration. In other words, you could call this 40 nanoequivalents per liter. So in other words, the free proton concentration in our blood is exceedingly small. It's in the nano equivalent level. All right. Now, small changes in the proton concentration result in large changes in pH. So it doesn't take a lot of proton for the pH to change. Okay. And so if our body is producing 50 to 100 milliequivalents per liter of proton a day, and remember, we're standardly, our level is going to be about 40 nanoequivalents per liter. 40 nanoequivalents per liter comes out to a pH of 7.4, all right? So if we're producing 50 to 100 milliequivalents per liter, that far exceeds the amount that would keep us at 7.4. In fact, since we metabolize and trend towards acidosis, that would make us extremely acidotic very quickly. And so in a case, of just normal metabolism, we trend towards acidosis, which would be an excess of those protons, and alkalosis would be a reduction in those free protons. And so our body's main job in healthy individuals at resting conditions is basically to make sure we can buffer these large amounts of acid that we produce from our metabolism. So buffers are extremely important. So their job is to resist changes in pH, and since it doesn't take a lot of proton to cause big changes in the pH, you have to think that if we're producing 50 to even 100 milliequivalents based on our diet and our metabolism, we're producing so much acid on a regular basis. We have to make sure that we have a lot of buffers, right? And so we have buffers inside our cell, outside our cells, and their job is to bind to those free protons, and that's going to resist those large changes in our pH, which could be deadly to our cells. So, for example, proteins like hemoglobin can bind to protons. We saw that in the respiratory lectures, where a proton can bind to hemoglobin and help to release oxygen, for example. And that helps to buffer the intracellular pH of red blood cells, for example. Phosphates, we talked about phosphates in the urine just now, so they can buffer the changes in pH in the extracellular fluid as well as in the intracellular. Bicarb is one of our main extracellular buffers, as I've mentioned. And bicarb is important because bicarb is something that we can produce with the kidney and regulate with our breathing. Okay, based on that formula that we talked about. Now, one really important key here is that buffers, although they resist changes in pH, they do not completely eliminate the change. Okay, so even if proton levels start to go up from our metabolism, it'll basically eliminate, or buffer 99.999 whatever percent of those free protons, but there'll still be a very small percentage of free protons, and that would still be enough to that can still be enough to trigger responses or changes in the pH. And this formula over here is just to show you this pH here, how it's calculated. So it's the negative log of the proton concentration, which, as you can see here, this is an equivalence. So you can see how small this is. Again, like 4 nano equivalents. Very, very tiny concentration, which keeps our pH at about 7.4. Okay, so you take the log of that, and you see it's going to be 7.4. Now, if we increase the proton concentration, right, the pH is going to go down. Okay, and if it goes, proton concentration goes down, pH goes up, and so on. So that's the relationship it has with the proton concentration. So, the respiratory regulates the CO2. So CO2 levels, which actually can change the way our buffers behave, like our bicarb buffering can behave. So bicarb and CO2 become, major players in our ability to regulate acid-base balance, simply because we can regulate them with our respiratory system and our renal system, and we can make adjustments in those two systems to help offset some of these changes. Right, so we know that breathing can change the CO2 levels, which can alter, the pH, and we also know that the kidney can excrete protons and produce new bicarb, and so this is also going to help us to readjust and get back to our normal pH. Okay, now I want to review the Henderson-Hasselbalch equation. This equation is actually useful in helping to represent the changes that are going to occur in our pH when our variables like bicarb or CO2 are altered. Now, the Henderson-Hasselbalch equation is really what it does is it takes a look at the ratio of acid and base to one another. And so that's what you're seeing over here. This is representative of our base, and this is a representative of our acid right here. Yes, I know in our formula here, CO2 plus H2O give us carbonic acid, that should be a 3. HCO3 minus plus the proton, so that's our formula. This is really our acid right here, our carbonic acid, but the carbonic acid is very, very small and it's difficult to measure. So instead, to substitute it, we know that there is 0.03 millimoles of carbonic acid for every millimeter of mercury of CO2. So instead, we multiply 0.03 times the CO2 as a substitute for our carbonic acid, which is difficult to measure. So CO2 becomes our substitute then. The 6.1, all right, for our formula, for our purposes here is a constant, which represents the pK, and the pK just simply represents the pH when both the acid and bases are in equal amounts to one another. So for example, if bicarb was equal to the amount of acid, which is the carbonic acid, if they're equal to each other, that would equal 1, that ratio. The log of 1 is 0. So our pH would equal 6.1. Again, that means the base and the acid were in equal amounts to one another. Okay. So this will be our constant for this particular formula. All right. And we are going to substitute, instead of carbonic acid, we're going to substitute PCO2 and multiply it by 0.03. So let's take a look at some real values. If the bicarb is 24 milliequivalents per liter, get rid of these arrows. So the normal amount of bicarb in our body is 24 milliequivalents per liter. A PCO2, let's say an arterial PCO2, that's about 40 millimeters of mercury. We're going to multiply that by the 0.03. So what that should come out to, 0.03 times 40 is 1.2 and 24. So it's 24 divided by 1.2, which is 20. Okay. So it comes out to 20. The log of 20 is 1.3. 6.1 plus 1.3 equals 7.4. So if you solve this, you're going to get a pH that's equal to 7.4. So this shows us that the ratio of bicarb to our acid, it should be 20 to 1. All right, so 24 milliequivalents of bicarb divided by 0.03 times 40, which is 1.2. That's a 20 to 1 ratio, which I show over here. So 20 times more base than acid, that's going to give us a pH of 7.4. And the reason I show you that ratio is because this helps us to explain when there's a change in the pH, how we're going to change the CO2 or the bicarb to adjust for that pH. So for example, let's get rid of some of the writing here. Let's use green instead. So let's say bicarb has gone up. Okay, so for whatever reason we have an increase in our bicarb. We can represent that down in our formula down here, an increase in the bicarb. All right, an increase in bicarb, it's in the numerator there, that's going to result in an increase in our pH. If we look at our formula, does that hold true? Well, yeah, if we increase our bicarb, more of that combines to the hydrogen ions, taking out more of those free hydrogen ions, shifting the equation in this direction. The loss of that free proton here is going to result in an increase in our pH. So this formula accounts for that, right? So that makes sense. Now let's see. Let's try one more example. What if the acid were to increase? Let's say CO2 is our representative of the acid in this case. So let's say the CO2 increases in the denominator there. And that's the only thing that changes. Well, let's see. If we have an increase in CO2, all right, an increase in CO2 is going to shift the equation in this direction, and we're going to produce more free protons, and so therefore will become more acidic. And in fact, that holds true in this formula as well, because if the numerator goes up and everything else holds steady, the pH is going to come down. Okay, so we can see that relationship here. Now, what about in terms of compensation? So let's try this example. let's go back to our increase in bicarb for a second. If I have an increase in bicarb, we know the pH is going to go up if that's the only thing that changes. Okay. Now if the pH goes up, well, if I want to keep my 20-to-1 ratio, and that'll bring you back to 7.4, in order to keep the 20-to-1 ratio, well, if I increase my acid, so I increase my CO2 as well, I can maintain my ratio. So if they go up proportionally. So that can increase my ratio and maintain my normal pH. It'll keep my 20-to-1 ratio, and I'll maintain that pH. So if we look down at our formula here, again, if the bicarb goes up, that shifts the formula this direction. Okay, that's going to shift this direction, which is going to cause a drop in my free protons, so my pH is going to go up. Any extra CO2 I produce, I'm really just going to readily breathe off with normal ventilation. So that's not going to accumulate. Okay. But if I need to compensate because my pH is dropping, that's going to be a strong trigger, okay, to actually hypoventilate. So in this case, I'm going to hypoventilate to increase my CO2. By increasing my CO2, that's going to help restabilize and push some of that back in this direction here and free up some of that proton again, bring my pH back to normal. Now we're going to go into some of the mechanisms of that, or some of the more detail on that in the slides to come, but this is the basic relationship here, is that ultimately, remembering this formula, you'll understand that as long as we can keep that 20-to-1 ratio between our base and our acid, we can offset that and make sure that we keep our pH normalized at 7.4. Okay, so we're going to keep that Henderson-Hasselbalch equation in mind, and we're going to use as an example to show you the importance of having an open system in regulating blood pH. And by an open system, I mean being able to regulate the pH via respiratory system and the renal system, and how that helps us to adjust our pH. So we're going to go through an example like that. All right. Now, first off, over here, I just want to point out that we have our equation, so we can look at that for reference. Okay. Now, in this example, we're going to start with what we call a closed system. And now this is theoretical. All right, we don't have a closed system. In fact, we're able to adjust CO2 and bicarb because of our renal and respiratory systems. But we're just going to take the example and just pretend right now that we have a closed system and I can't breathe off that CO2, for example. And let's see what happens if I add 10 millimoles of a strong acid. Okay, now if I add 10 millimoles of a strong acid, bicarb is my buffer. So remember, we're starting out normally, bicarb is 24 milliequivalents. The CO2 is 1.2, so that's the 0.03 times 40, which we went over in the previous slide. And if we plug that into our Henderson-Hasselbalch equation, that comes out to a pH of 7.4. Okay, so that's all our standard conditions there. Now we add 10 millimoles of our strong acid. When you add 10 millimoles of a strong acid, you're really essentially adding a large increase in the amount of free protons. And if we what's going to happen is the bicarb is then going to bind to it because that's the buffer. And so we're going to lose 10 millimoles of bicarb to bind to this very strong acid. Okay, so when it combines with that, we're going to lose the bicarb when it joins that, and it's going to shift the equation to the left in this direction. So 10 millimoles, again, we started with 24 milliequivalents, and in case of bicarb, remember, milliequivalents and millimoles are the same in this case. So we're basically we're losing 10 milliequivalents of bicarb. So 24 minus 10, that comes out to 14. So that's why there's a drop right there. And our CO2 should increase because we shifted to the left. And that should increase by the equivalent amount of 10 millimoles of CO2. Right? So that's going to go up to 11.2. All right. Now, so that goes up to 11.2, so it goes up proportionally. And if we plug that those new numbers into the Henderson-Hasselbalch equation, you see 14 now divided by 11.2. And those numbers, again, were because of that shift. The pH is going to come out to 6.2. And that is a very precipitous drop in our pH and can be lethal. Okay, so a drop like that can be lethal. And the only reason we'd have a drop like that is because we didn't get rid of the CO2. Right, the CO2 increased. So this is a hypothetical situation wherein that CO2 increases proportionally to the amount of bicarb that has been decreased. But this is not what happens in our bodies. What we actually have what we call the open system. This is the importance of this open system down here, Instead, we remove the extra CO2 when we breathe it off. Now, this is not hyperventilation. This is simply the extra increase in CO2. We will simply ventilate it off. Normal ventilation will get rid of any excess CO2. So let's take a look. The bicarb will still drop. So let's say it drops to 14 again. But instead of getting an increase in CO2 to 10, it stays normal. It stays at 1.2. Again, the reason is because we just exhale off the extra CO2 during normal breathing. Does that help the pH? So if we take a look, if we plug those new numbers, 14 divided by 1.2, our pH now comes to 7.17. So yeah, we don't have as big a drop because we're able to get rid of that CO2. Okay. Now finally, this is where you'll see that we're not at 7.4 yet. We're still acidotic. So this acidosis, all right, this acidosis is going to be a strong stimulus to our chemoreceptors in our brain to trigger us to hyperventilate now. So this is where you're going to see hyperventilation kick in. This is now going to be the respiratory compensation. So we hyperventilate, and what that's going to do is it's going to drop our CO2 even further. So let's say instead of being at 40 millimeters of mercury, we drop it to 30 millimeters of mercury. Multiply that by 0.03, that's going to come out to 0.9. So if we plug in 14 divided by 0.9, we're going to get a pH of 7.29. So what happens here is not only to just regularly blowing off that extra CO2 help improve our pH, but then we compensate by hyperventilating and blowing off more CO2. This actually helps produce a better ratio between our base and our acid and brought our pH back up to about 7.29, which is much better. And then finally, the kidneys. The kidneys are slower than the respiratory system in terms of compensation, but when they kick in, they're going to add new bicarb to the blood and get rid of the extra protons, which like we discussed in the earlier slides. So they'll reabsorb the bicarb, they'll produce new bicarb when they secrete that proton, and so that's going to bring the bicarb back to a normal level. And when the bicarb comes back to the normal level, the pH starts to come up as well, and our respiratory rate will actually start to normalize, and we'll come back to a pH of 7.4. So this is sort of demonstrating to you now the role that having the lungs play in just getting rid of excess CO2 and then actually stimulated to hyperventilate to help maintain that ratio. Because I talked on the previous slide that we're trying to maintain a ratio of 20:1 in terms of base to acid in our formula there. So if we're looking at it in terms of our Henderson-Hasselbalch equation, what happened was we had a drop, right? We had a drop in our bicarb, and to compensate for that, we started to drop the acid as well to keep the ratios as close to possible to maintain our pH. The way we were able to do that was through hyperventilation and ultimately replacing that bicarb. So our respiratory system is going to regulate our PCO2. It's going to be able to compensate for metabolic causes of acid-base changes, just kind of like I described in the previous slide. It is considered what we call our second line of defense, just behind the buffers. The buffers react first to any acid-base changes. So that's our first line of defense. So that's going to minimize changes in pH right there. And then our second line will be the respiratory system to help offset those changes as well. And the renal system will work on the bicarb and the hydrogen ions. Okay, it'll regulate those levels of bicarb and protons in order to adjust for acid-base disturbances as well. So we have both the CO2. So the order of operations will really be buffers first, they act the fastest, then the CO2 via the respiratory system, and then the renal system. The renal system is a good bit slower and can actually take several days to have significant impact. So I want to use this graph now called the Davenport diagram to help us understand the process of what happens with acid-base dysregulation and how our body compensates for that. And so we're going to use the same variables, or we're going to be looking at the same variables that we saw that we use in the Henderson-Hasselbalch equation. So for example, we have bicarb here on the y-axis, and that's in milliequivalents per liter. And again, we talked about that being normal at 24 milliequivalents per liter. Our normal pH is 7.4, which you see there on the x-axis. You see these curves here. You see three parallel curves. These are the PCO2. So they represent our PCO2 in millimeters of mercury. So 40 would be our normal. Less than 40 would be like hyperventilating. greater than 40 would be like hypoventilating. And so where the PCO2 of 40, bicarb of 24, and our pH of 7.4 all crisscross right here in the center. So that's our starting point, our normal. This line that's drawn through them right here, over here, this is called the buffer line. And this buffer line represents all the other buffers in our body besides bicarb. And basically it has the slope that it has because all these other buffers do play a role. When we have an increase in acid production, these other buffers are going to help to resist that change in pH along with bicarb. So we're going to account for this action of other buffers by this particular line, which in this case is going to be a constant. Okay, that particular slope is going to be our constant. And we're going to see how bicarb in particular is affected. Okay. Now, if we started our middle point right there that I've dotted there for you in red, and let's say we're now in a state of metabolic acidosis. Now this is a pathological condition. This is not like the normal amount of acids that we produce when we normally metabolize. Okay, so this is beyond that level. So we're producing extra acids, and in this case, okay, this could be coming from lactic acid acidosis. This could be coming from keto acids. There's just a multitude of different causes for metabolic acidosis. The idea here, though, is that the insult happens first, and that's going to decrease my pH, right? Because I have increased acids. Now, when I increase the amount of acids that my body is producing, that's going to cause a drop in my bicarb. So I'm going to draw a little down arrow here by the bicarb. Okay, so this is a drop in my bicarb, and this is what I refer to as really the insult. This is what's causing the problem. All right, I have more acid, so more of my bicarb is dropping to buffer that acid. So the bicarb level should also drop. And in fact, we haven't changed our rate of breathing at this point. So we follow that PCO2 curve, all right, down in this direction. I'm going to draw a new point, let's say right about here. So again, following down this curve like that, all right? So right to about that point. So what's happening is we're becoming more acidotic and our bicarb level is dropping. All right, so this quadrant down here represents our metabolic acidosis. Now this is not compensated for yet. So I want to bring my pH back to 7.4. So in order to do that, I have to have some sort of compensation occurring here. So first, let me label this dot right here as A. So A, all right, that represents uncompensated. All right, uncompensated acidosis. And there's a decrease in my bicarb, which was one of my initial insults, right? And that's going to ultimately lead to a decrease in my pH because I have all these extra acids. So I've dropped my pH and I've dropped my bicarb. So this is how I'm acidotic, and I see my bicarb levels are low because we're trying to buffer those extra acids. So I'm going to draw another line right to here. Now we're going to move in this direction towards, I'm going to call it point B here. Now point B, I drew the line, and this line right here is parallel to my buffer line because there are some buffers that are going to be other buffers that are going to help us out there. So it's going to follow parallel to that slope right there. But you'll notice what's happening here is this. I'm shifting curves to now the decreased, all right, PCO2 of less than 40. What that means is that I'm actually hyperventilating. So I'm going to start to blow off that extra CO2. When I blow off that extra CO2, that's going to cause a further shift of this equation in this direction. That's also why you see the bicarb drops a little bit further as well, down to B there, down to point B. And my PCO2 is low because I'm hyperventilating. So B, this is respiratory compensation. Now, respiratory compensation, where you're going to see a decrease in the PCO2. All right, and now the decrease in PCO2 is because I have an increase in my ventilation. And so this represents, that compensation. Now it didn't go all the way back to 7.4 because the problem is this. As I start to breathe off more and more of that CO2, and my CO2 level starts to drop, that actually suppresses my ability to hyperventilate any further, because decreased CO2 inhibits any further hyperventilation. So it limits me from being able to restore it all the way. Now, in order to restore myself back to normal, I'm going to draw a little zigzag line right here, back to point, let's say C. Well, at point C, the only way I can restore myself back to normal is I have to recover my bicarb, right? So I need renal compensation at this point. And that kicks in, a couple of days later, but I get renal compensation, and that's going to excrete protons and reabsorb bicarb. And ultimately, all right, this can help me recover. Now, the only way I can fully recover, though, and this is key, is that I still have to treat the underlying disorder. So I'm still trying to figure out what's going on here. So just to recap, right? If I started out at point C here, which is my normal, I went to point A. That was my uncompensated state. I'm becoming acidotic, and my bicarb is dropping because I'm producing all these extra acids that need to be buffered. All right, that's going to shift the equation to the right, as you see at the bottom there, and then I'm going to compensate to point B. So I'm going to increase my respiratory rate, and I'm going to drop my CO2 level. All right, and that's going to shift it even further to the right, but the idea is here that I'm going to also be removing free protons from that solution to help me offset this and bring my pHs close to normal. But I can't completely recover just from the respiratory compensation, because I'm not replacing the bicarb or getting rid of any protons, and by hyperventilating, I'm dropping my CO2, and my chemoreceptors are going to inhibit me from dropping my CO2 any further. So I'm stuck at point B until my kidneys kick in and can help me reabsorb some bicarb and excrete that proton. And they will help me get even closer to 7.4, but I can't get all the way back to my starting position unless I treat that disorder. Otherwise, I'm going to continually be producing too many acids. So if I was going to diagnose metabolic acidosis, look for a drop in the pH, a drop in bicarb. So those two tell me it's metabolic acidosis, and my compensation is a reduced PCO2. Now, anytime we have a metabolic acidosis, we have to consider what we call an anion gap. An anion gap is just simply something that we use to help us make up a differential for what might be causing metabolic acidosis. And there are, many different things. So what we do is we actually calculate it using sodium chloride and bicarb. We take those concentrations in the plasma. We actually sum up the sum of the cations and anions are equal in any body fluid, because we have electrical neutrality in our extracellular fluid there. So the number of cations and anions should equal one another. So the formula is basically we calculate sodium concentration, and that makes up the vast majority of our cations. But then there's going to be some unmeasured ones that we're not really accounting for. And then chloride and bicarb make up the vast majority of our anions, but there's also unmeasured anions. But they should all equal each other when we're electrically neutral. So the unmeasured anions is our anion gap. So what we usually do is we don't really pay attention too much to the unmeasured cations, because they're essentially so few in number relative to sodium. So we just say sodium minus the chloride plus the bicarb. And so there is going to be a difference. So what happens is sodium, let's say, is 140. 140 minus 105 plus 24. Okay, and that's going to give us 129. So we're going to have an anion gap of 11. So this is what we actually take this and measure this. We get the blood labs, take the sodium, chloride, bicarb. All right, and you're subtracting it, and so the 11 milliequivalents per liter. So what this means is that there's a difference in our cations and our anions that are unaccounted for, and that difference is about 11. And so we have a normal difference because we don't always just measure every single anion that we have, because they're numerous, even though they're in small amounts. And so we know that there's a normal gap that we have there. All right, and that normal gap is particularly between 8 and 14 milliequivalents per liter. So 11 is right in that normal range that we know that there's a difference. However, if we get an acid like lactic acid, for example, all right, and lactic acid is added to the plasma, bicarb will decrease, right, and chloride is not changed, all right, then the anion gap will increase. So for example, if bicarb decreases and chloride doesn't change, all right, so when I add these two together, it's going to be an even larger gap. And so we call this, an anion gap acidosis, and there's certain acids that can do that. All right, the unmeasured anion in this case is the lactate. Now, in normal anion gap metabolic acidosis, okay, this usually occurs with a loss of bicarb again, like diarrhea, for example. But we have exchangers, bicarb chloride exchangers, for example, and we'll get an equal exchange of chloride for all the lost bicarb. So the chloride is replacing the lost bicarb. So what ends up happening is, since it's replacing the loss of all the bicarb, it maintains the gap. Okay, so some acidosis will end up with chloride that replaces the bicarb and maintains the gap. In other types of acidosis, we'll end up with a larger gap because it's going to decrease bicarb without any compensation or any increase in the chloride. So later on, you guys will learn in the pathophysiology lectures how what the differentials are for, say, metabolic acidosis with an anion gap versus a normal anion gap. Now, metabolic alkalosis. Again, here's our starting point, right here in the center on our Davenport diagram. And so, if this is now alkalosis. So in alkalosis, we are going to be shifting in this direction here because we're becoming more alkalotic. Now in alkalosis, we end up with essentially more bicarb. Okay, where there's a drop in the amount of protons we have. Like for example, in vomiting, we vomit, we get rid of excess acid, but we're leaving behind bicarb in our blood, so our bicarb level can be elevated. Or they could be ingesting something that is very alkaline, which is increasing their bicarb concentrations. Now in this case, again, there's going to be a shift of the pH that's going to go higher, and the bicarb levels are going to elevate. So we're going to be in this upper right quadrant here. Remember, there's no change in our respiratory rate initially, but the insult is that we are getting an increase in our bicarb. So we're going to get something that looks like this, let's say. We'll draw a point here, so we'll make that our point A. So point A over here, that's going to be our uncompensated alkalosis. And again, this is where the pH is going to be elevated and the bicarb is also increased. All right, so again, having an elevated bicarb is like adding base. If you remember the Henderson-Hasselbalch equation, an increase in bicarb will increase our pH. So this is the uncompensated. Now to compensate, remember we want to go back to 7.4. So to compensate, we actually have to hypoventilate or decrease our ventilation. And that in turn is going to increase our CO2 levels. So this is the compensation down here, and it's increasing the CO2. So that means we're going to shift in this direction over here to a greater PCO2. I'll make that point B. So point B is respiratory compensation. Okay, that's going to be an increase in our PCO2. All right, that means decrease in ventilation. So we hypoventilate, increase our CO2. That helps to push back the equation in this direction. All right, so when increase that pushes it in this direction, that helps to increase the number of free protons, which helps to decrease our pH back into the normal range. Okay, and again, this is going to be essentially self-limited because as the CO2 starts to accumulate, we're going to want to start to hyperventilate. So we end up kind of self-limiting there at that point. That's sort of fixed at this point to maintain a pH that's not quite at 7.4 yet. Now finally, in order for us to kind of zigzag our way back to point C here, in C we need, again, really the main issue is we have to correct, all right, correct the underlying condition. But we're going to get some renal compensation there, right? So the renal system is going to excrete bicarb. All right. So this is going to happen because we're going to increase the amount of bicarb that we filter because we're in metabolic alkalosis. So an increase in bicarb filtered is going to result in more bicarb loss in our urine, as well as something called the beta intercalated cells. I'm going to put IC here, beta intercalated cells. We talked about alpha, but beta intercalated cells are basically the opposite of alpha intercalated cells. The job of the alpha intercalated cell is to secrete protons and reabsorb bicarb. Beta intercalated cells have the opposite polarity in the collecting ducts. What they'll do is they'll actually secrete or excrete bicarb and reabsorb protons. So these help to secrete bicarb. For every bicarb that's being secreted, we can reabsorb some of the proton as well, and that can help bring that pH down to our normal level. So this is how the renal system would compensate. But again, it can only get us back to our normal if we, help to, or if we can make an effort to correct the underlying condition. So over here, in metabolic alkalosis, look for an elevated pH, an elevated bicarb, because that's the initial insult, and then the compensation was going to be an elevated PCO2 to help bring that pH back down. For respiratory acidosis. Now, respiration is the initial insult in this case. So down here, that means we're going to have an increase in our CO2. So I'm going to call this side is now the insult. So something's wrong with our respiratory system where we're retaining CO2. So if we're retaining CO2, if this is our normal starting point, we're going to shift along this buffer line curve right here to point A over here. And you'll notice we're retaining CO2. That means our PCO2 is going to be greater than 40. That's going to shift the equation in this direction. We're going to produce more protons. So this is why we're becoming acidotic. All right, we're dropping our pH. Because we're producing protons, we're also producing equal molar amounts of bicarb. So bicarb level does go up as well. Okay, so bicarb level is elevated. Now to compensate, I'm going to draw a line to here, where our respiratory rate is still where it is, which is the initial insult. But this is where we're going to have renal compensation. So first, let me put down point A. Point A is uncompensated. We have an increase in our PCO2, a decrease in our pH. All right, because we're in acidosis and the CO2 level is what's causing that. At point B, this is renal compensation at this point. Because remember, we cannot compensate with our respiratory system. In other words, the respiratory system cannot compensate for a respiratory disorder like acidosis or alkalosis because it's the system that's causing the problem. So renal compensation will kick in, but again, this takes time. This could take 3 to 5 days even. And you're going to see an increase in the bicarb. Right? So you see the level of bicarb goes up a little bit further, but this is because we're reabsorbing the bicarb and producing new bicarb in order to buffer that acidosis. At the same time, we're going to drop the proton level because we are going to be excreting it to help drop that, to bring that pH back up. Now, in order for us to correct back to normal, right, to get to point C again, well, to get to C, it's the same thing as we talked about before, which is correct the problem or correct the disorder. Whatever the pulmonary disorder is, you have to do your best to kind of correct that or medicate it or what have you in order to correct that so that the CO2 will actually renormalize. Okay? Otherwise, you're going to stay in sort of that renal compensation state. All right, so again here, in terms of our formula, remember the insult is the increase in CO2, the renal compensation over here. So this is renal compensation would be to increase that bicarb level in the hopes of being able to combine with more of that proton and shift back into this direction here. Okay, so we can bring that pH back up. Respiratory alkalosis. So in respiratory alkalosis, we're going to go into the bottom right quadrant now. And so we're going to be moving from this point here along our buffer line to point A. So this, again, respiratory system is what's causing the problem in the first place. But in this case, our PCO2 is lower because we might be hyperventilating, for example. And so we're dropping our CO2. Now we drop our CO2, all right, this causes the shift in this direction. So more protons combine with bicarb, so that free proton level goes down, and so the pH goes up. And so that's why you see the pH going up here. The bicarb level is also going down in this shift, so that's why you see a shift in the bicarb that's going down here to point A. So point A represents our uncompensated state. We have a decrease in our PCO2 and an increase in our pH. Now point B. Point B is going to be somewhere over here. All right. Now point B. So point B, you see that we lose a little bit more bicarb there because this is what's happening here. We have renal compensation. All right, so renal compensation, we're going to have a decrease in our bicarb level. All right, we're going to get rid of that extra base, so we're going to excrete that. And we're going to increase in our proton level, all right, because we're going to reabsorb some of that proton. So now that's going to help us bring our pH back towards normal, all right, to drop our pH again. However, again, we're not fully corrected until we correct the underlying disorder. So again, treat the disorder in this case, whatever the particular lung problem happens to be, or whatever is causing this increase in ventilation, you're going to treat that first, okay? Or treat that, not first necessarily, but treat that in order to help correct things back to our normal functioning level. So we're going to have again, to diagnose it, you have an elevated pH, all right, because we're alkalosis, reduced PCO2, because that's the initial insult. Down here, that's the insult. The compensation is to reduce the bicarb. So the bicarb level will come down. A dropping of the bicarb when we compensate. So this is the compensation. Dropping the bicarb is going to help shift things back in this direction to help us increase that proton level, which helps to drop our pH from being too alkalotic. So that's caused by the renal system. This is showing you a summary of the things I just talked about. So that's acidosis versus alkalosis in each one of those quadrants. So you'll notice here we started with metabolic acidosis, and in the uncompensated state, okay, this is our PCO2 of 40. To get compensated, we try to shift towards the pH of 7.4. So that's our goal in compensating, right? And then you see over here, the metabolic alkalosis up in this quadrant over here. Here's our uncompensated state, and we shift in this direction towards 7.4 in order to compensate. Respiratory acidosis over here, we're uncompensated, and then we shift in this direction back towards 7.4 in order to compensate renally, for example. And then the last one, here's our uncompensated state, the one we just talked about, respiratory alkalosis, and shifting back to 7.4. So you see the shift is always back towards that midline to try to readjust and get that pH back to within that normal limit there. Okay, so everything I've just described, respiratory acidosis and alkalosis, metabolic acidosis and alkalosis, if the patient only is exhibiting one of them, so just respiratory acidosis or just respiratory alkalosis, and they're also showing the appropriate compensation, we refer to this as a simple acid-base disorder. However, if there's not an appropriate acid-base compensation, there might be a mixed disorder going on. So in other words, there's a predictable compensation that occurs with each one of these disorders. And if we see that predictable compensation, we call it simple because we know that it's strictly due to just respiratory acidosis or respiratory alkalosis, and we see the appropriate compensation taking place. But sometimes you can have what we call mixed acid-base disorders, where somebody could have a respiratory issue as well as a metabolic issue. And so the compensations aren't quite as clear cut. So we haven't really talked about those sort of complex situations. You'll get more into that later on. But this flowchart helps us to at least analyze simple acid-base disorders. And if you can recognize a simple acid-base disorder and you see that there may not be the appropriate compensation, then that might lean you to thinking that this could be more of a mixed acid-base disorder. Plus the history and so on will help to kind of elucidate some of those issues as well. But let's just go through the flow chart for the conditions we just talked about, all those simple acid-base disorders. So the first thing is we need to get an arterial blood sample, right? We need to get that ABG. We get the ABG, and the ABG, it can measure a pH, a bicarb, and a PCO2. So the first things first is look at the pH. Are they acidotic or alkalotic? Let's say they're acidotic. All right, well, the next step is look at the bicarb and the PCO2. So for example, let's say we're looking at the PCO2, and the PCO2 is elevated. Well, an elevated PCO2 can cause acidosis. Okay, right? So over here, an increase in CO2 causes a shift in the equation, so we're increasing the amount of free proton. That can cause an acidosis. So if we see that the level of CO2, all right, is consistent with the pH, then it's a respiratory issue. Okay, so an elevated PCO2 is consistent with being acidotic, and the person is acidotic, so it's a respiratory acidosis. Well, what is the compensation then? Well, if you take a look at the bicarb, if they're compensating appropriately, the bicarb levels should start to elevate. Remember, the kidney should start to reabsorb and produce new bicarb in order to help shift the equation back again in order to produce, or in order to drop that free proton level to compensate, all right, to recover. So this is the renal compensation for that. So if you see somebody's acidotic, they have an elevated PCO2 and an elevated bicarb, then that suggests that this is a respiratory acidosis. Now, let's say they're acidotic, but the PCO2 is actually not elevated, it's low. Well, a low CO2, all right, a low PCO2 does not cause acidosis, so that doesn't actually work together. So if it's a low CO2, that would actually cause a shift in this direction. The person would actually become alkalotic. So that actually makes more sense for a respiratory alkalosis, but this person's not alkalotic, they're acidotic. So then you look at the bicarb, and you see the bicarb is low. Well, the bicarb being low makes sense for a metabolic acidosis because the bicarb gets used up with this overproduction of acids. So the bicarb initially drops. That's the initial insult in metabolic acidosis. So a low bicarb. So the low CO2 can be explained because the low CO2 is a compensation for the metabolic acidosis. By dropping the CO2 level, okay, by dropping that CO2 level, I can actually decrease the amount of free proton there and shift that equation. So in doing that, that allows me to readjust my pH. So in this case, all right, so the person is hyperventilating in this case. They're blowing off that extra CO2 in order to compensate for a metabolic acidosis. And this is something you can actually see within the patients, for example, in diabetic ketoacidosis, for example. That's a metabolic acid disorder, and they often breathe very quickly, all right? And that hyperventilation is to blow off the CO2 in order to change their pH back to, or increase their pH back to normal. And of course, the whole thing works like this. If you go to the alkalosis, if they're alkalotic, if they have a decreased PCO2, that's consistent with a respiratory alkalosis, all right? And then that means the bicarb should be reduced. And if you see that the bicarb is elevated and they're alkalotic, well, that's consistent with a metabolic alkalosis. And then you take a look at the PCO2 to see if they're compensating by increasing their partial pressure of CO2. So we have to look at all those in order to make the diagnosis and see if in fact they're compensating. So this is an example I'm going to go over with you guys in the Zoom session. But I want you to take a look at these different ABGs and see if you can figure out which ones are normal and which of these simple acid-base disorders are which, and if they're if they're compensating. Okay. And this is another case that I'll go over with you guys in the Zoom session here. All right. So this is a patient with a history of emphysema, admitted for severe diarrhea. And then I'm giving you the following ABG, and I'm also giving you an anion gap. So we'll be able to kind of go over that and interpret that in the Zoom session.